All right, so we've gone into how enzymes work. We've gone into how to speed up reactions. Now we need to get into how those reactions happen, where they happen, and what's going on in the cell. So we're going to talk about cellular metabolism. All of the reactions building things up and breaking them down. Cellular metabolism is the combination of all of the cellular processes across the board happening in the cell. Here is a basic cellular pathway of the breakdown of glucose. You're going, let's see if I can figure this out. Let's see, So right here we have glycolysis. So we're going to start breaking down glucose here. It's going to produce these byproducts. Those are going to be broken down. You'll notice that each of these steps has a little blue line moving from one thing to another. So here you have a reactant that becomes a product. It's a process. Each of those blue lines represents an enzymatic reaction. Specific enzymes are involved. And they're very different enzymes for each. It's almost like a flow chart. You guys ever worked with flow charts before? One leads to the next, to the next, to the next. That's what's happening here. A reactant leads to a product. Now that product is the reactant in another reaction. Then that product is a reactant in another reaction. On and on and on. So what we're looking at is now all of these reactions occurring in the cell. And this looks complicated. The thing is, a scientist could take their entire career just to figure out this one small section. And then call their life well worth it. That is, it seems like a small piece, but there's a lot of work that goes into figuring out each of those little steps. So as scientists work together, they build a more complete picture. Now this is one reaction happening in a cell, one group of reactions happening in a cell. This is not all of the reactions happening in the cell. That last slide was just this little piece right here.
That's crazy.
Pardon? That's crazy? And so we're mapping out the slide, and that it gets even crazier because this is just 1 out of 47 pages that looks like this to map out all of the reactions happening in the simplest cell. Not even in your cells. So it gets complicated very, very quickly. But in the end, it's all just very simple pathways following the flow charts. This is the breakdown of lactose. You guys have heard of lactose before, right? Where have you heard of lactose? You hear of lactose intolerance. People can't drink milk. Right. Thanks. Lactose is broken down by what's called an enzyme called beta-galactosidase. So that lactose, using an enzyme, and is broken down into galactose and glucose. That glucose can bind with ATP. And when it does, it gets an energetic phosphate attached to it, so it becomes glucose 1P, 1-phosphate. That enters into a process called glycolysis that then breaks down that glucose. Going in the other direction, we have galactose gets broken and gets phosphate added to it to become galactose 1P. That then becomes UTP galactose to become UDP glucose to become glucose 1P, and then it enters in. It's just step by step following the flow chart. Can you skip steps? No, not really. If you skip steps, bad things happen. You can follow in a different route, but if you skip the steps, then galactose 1P cannot become UDP glucose, which means you don't get glucose 1P. I will not ask you to memorize a pathway. What I'm asking you to understand is the idea behind it. On the next test, you're going to memorize this pathway in much, much greater detail. For the next test, not the one coming up, the one on cellular respiration, we're going to cover it next week. In fact, it's the, I believe, yeah, it's the next lecture.
So after this lecture, can we stop setting the exam?
Yeah, this is the, it's a test. We don't want to freak Reagan out. But yeah, this is the last test that will be on this coming, I'm sorry, this is the last lecture that will be on the coming lecture.
Lecture on Wednesday next week after the test? No lab?
I don't know. I'll have to check. So we're going to talk about two types of reactions, and you know them, anabolic reactions and catabolic reactions. We're going to start with the catabolic reactions. Basically, give an example of a catabolic reaction. You destroy things because they're right, cats. Cats destroy stuff. One example. What chemical reaction breaks things down? Hydrolysis. If it was condensation, I would have asked Sarah. All right, so we're going to, the reason we have these catabolic reactions is several-fold. One big reason to have catabolic reactions is for recycling. It's way easier to reuse pre-built components. Remember, all of our macromolecules are made up of these individual subcomponents. What are those subcomponents called? What are macromolecules in general made of? Monomers. Monomers come together to form these polymers. Each of those monomers takes energy to build. But if we can recover them, we don't have to invest the energy in rebuilding new ones. If I wanted to build a protein, I could start from scratch and build all of my amino acids. Or I could take another protein that's not doing anything anymore and break it apart and rebuild it. Okay, Legos. You guys have, I assume, had a childhood where you at least played with Legos once in your life. If you played with Legos, you build something. You might follow the directions and build that thing that came with the box. What do you do after that? You do your own thing. Well, you can, but the thing is, you don't want to, you got this one thing you've already built. You're not playing with it anymore, so you can take it apart and build something new. You don't have to invest resources in building those the new subunits. Now, what would happen if you really, really liked whatever you built at first? What would you have to do? You'd have to buy new ones. You'd have to invest energy. You're right, put it on a shelf, set it aside, but then you have to go buy new ones. You have to invest energy. You have to invest resources. So it's easier just to break down something that's pre-existing that you're not using anymore to build new components. So that's really what these catabolic reactions are used for, recycling macromolecules. So what is, we've got a bunch of macromolecules, right? What are the monomers of the nucleic acid? Nucleotide. What are the monomers of the proteins? Amino acids. What's the monomer of the polysaccharides? Monosaccharides. Well done. By breaking down these larger molecules, you can get the components to rebuild new large molecules. The other thing, it's not just about recycling, it's about recovering stored energy. When you build bonds, you store energy. When you break bonds, you release it. So they're going to be, if you're releasing energy, it's an exergonic reaction. The energy produced from breaking down those molecules then can get stored in what are termed energy intermediaries, little batteries. Now, we've already talked about one energy intermediary. What was that major energy intermediary that we talked about? We talked about one energy intermediary, one cellular battery, one molecule that we build. Yeah, we have to recharge it, but what is that molecule? I know. Adenosine triphosphate, ATP. Well done. ATP. So it's the next slide, I know, right?
That rhymes. ATP, the rechargeable battery.
Haha, that you can remember it. So ATP can be hydrolyzed. You're going to remove a phosphate, releasing energy. That's a catabolic reaction. Or it can store energy by being built. It can store energy as an endergonic reaction, a condensation reaction, to go from adenosine diphosphate to adenosine triphosphate. There are two broad mechanisms for building adenosine triphosphate: substrate level phosphorylation and chemiosmosis. Substrate level phosphorylation is where you use an enzyme to directly build adenosine triphosphate. You take an adenosine diphosphate and a phosphate and you squish them together. Diphosphate and a phosphate, you squish them together, and now you have triphosphate. So that's it. Substrate level phosphorylation is where you use an enzyme to build ATP. That is the whole definition. So when I inevitably ask you, what's the difference between substrate level phosphorylation and chemiosmosis, part of that answer is substrate level phosphorylation requires an enzyme. All right? Substrate level phosphorylation and chemiosmosis. So the way I remember that is we see substrate. When you see substrate, automatically think enzymes. Enzymes go with substrates. And phosphorylation means adding a phosphate. So that's the easier one. Substrate level phosphorylation requires an enzyme to build ATP. The other mechanism for producing ATP is called chemiosmosis. Chemiosmosis is where you're going to use the energy stored in an electrochemical gradient to make ATP. Okay, now we've got to remember what a gradient is. So what's a gradient? Solute? Yes. Solute will follow its concentration gradient. So if you have a high level of solute in this space here, it's going to want to get into that space there. It wants to go from high to low concentration. It wants to diffuse. Make sense? So what we do in our mitochondria, aerobic cellular respiration, how we produce ATP, is we're going to create a very, very high gradient in that space, in that intermembrane space. Because remember, mitochondria are double membrane bound, an inner membrane and an outer membrane. So between the two membranes, you have a lot of hydrogen ions, tons of them. Those hydrogen ions want to get into that matrix. They want to get on the inside. The only way they can get through is through that molecule there called ATP synthase. ATP synthase is a channel. It will let these hydrogen ions through. But as they go through, they spin it. They make it rotate. The energy, the kinetic energy of that rotation is then translated or transferred to take an adenosine diphosphate and a phosphate and squish them together. So the idea is, you guys know what a water wheel is? Okay, a water wheel, as it spins, you've got river water running like, river water's going this way, so the water wheel is spinning like that. Okay? That kinetic energy can do something in a wheelhouse, whether it's hydroelectric power or whatever. What the ATP synthase is doing is it's spinning just like that water wheel, and it's turning that energy into the bonds of ATP. That's the simple explanation. We're going to get into this in a lot more detail in the lecture on cellular respiration. It may make more sense then. I just wanted to introduce you to the concept because this plays a major role in both photosynthesis and aerobic cellular respiration.
So are we going to have to know everything you told us?
You need to know that. Yeah, no. Simple definition for this particular test. For the next test, I will expect you to explain it in detail, probably as an essay.
What's the essay you need for this test?
I haven't written it. No essay? Let's see, what have we talked about? Oh,
Word bank.
we'll talk.
Word bank. Actually, you said that one, I think it's going to be describe the process of protein. Or the cell.
Oh, well, that's going to be the way a protein is embedded in the membrane. There's probably going to be a short answer for 3 points.
What about like the cell theory?
Cell theory, 3 points. Yeah, I'll figure it out. I'll have it written by next time we meet. Okay, so we need to move energy from one place to another. We have to create this adenosine triphosphate. You have to understand with energy, what we're dealing with ultimately is the movement of electrons. What happens when an atom absorbs energy? Not really, but the electron cloud expands because those electrons are moving faster. Now, if you took those energetic electrons, those electrons that are moving really fast, you can move them from one molecule to another. And then they could move from this molecule where they were doing something take that energy with them to another molecule and do something new. Just like when you take the remotes, the batteries out of your remote to power something else, your remote no longer has power, but you have something else that can do work. So this moving electrons, if you were going to lose an electron, an electron is lost. That's called oxidation. If you receive an electron, that you take that electron in, that's called reduction. And there's some good reasons for that. Because it really, on its surface, doesn't make much sense when you're adding an electron, you're gaining an electron, you're reducing. But the thing is, think about the charges. If you add an electron, what charge does an electron have? Negative. So if you add a negative charge, what happens to the charge?
More negative.
Yes, the charge goes down. The charge is reduced. That's where that word comes from. With oxidation, oxidation is all about losing electrons. All right, what do you guys know about oxygen? We breathe it. It has a very high what?
Polar.
That's water. But it's the property of oxygen that makes water polar. Has a high what? It starts with an E. High electronegativity. So it pulls electrons to itself, right? So if oxygen's coming by this sodium here, it will steal electrons. So that sodium got oxidized. Yes, this is stupid, but check this out. So it's like, let's say this is an electron, right? It's hanging out by you. You're sodium, I'm oxygen. I come through and I'm like, oh, you've been oxidized. The way I remember is Leo goes grr. Leo being lion, right? So lose electrons, oxidation. Leo, lose electrons, oxidation. Gain electrons, reduction. Grr. Because I have to gauge where the class is based on where you are. I figure out, like, I look around. So in case you guys didn't know, when I look at different people, I'm gauging where the class is. So you're one of the people I look at. I look in the back. I check Gabby. If you're on your phone, then I've lost everyone. It's like, that's why I look at different people. No problem. You're one of the ones I look at. I check out Sarah, too. She's right there. Annie tends to know it, so I don't have to worry about her. Oh, no. I am really proud of you. Like, you're blowing this thing away. Lose electrons, oxidation. Gain electrons, reduction. Now, if you've been in chemistry, you may have heard of another one. I don't care how you remember it. You just have to remember it. Chemistry, they like to say, like, oil rig, because it's oxidation is loss, reduction is gain. Yeah. Which one? Leo goes grr. L-E-O. So lose electrons, oxidation. And then grr is gain electrons, reduction. Leo goes grr. Gain electrons, reduction. And moving energy through a system is all about the oxidation reduction reactions. This is oftentimes shortened to say redox reactions. So because oxidation reactions will always go with reduction reactions. One doesn't happen without the other. They're combined. They connect. Where one atom loses electrons, another necessarily gains them. We have a bunch of examples in nature. One is rust. If you were to leave iron outside, what happens to it? It turns to rust. It goes from being one particular chemical state to another, because oxygen's coming along and stealing electrons. In the Krebs cycle, the Calvin-Benson cycle, which we'll get to in photosynthesis, an entire stage is devoted to reduction. But then we have, this is one of my favorites, the Statue of Liberty. What's the Statue of Liberty made of? What color's copper? What color is the Statue of Liberty?
Green.
Why? Oxidation.
Oh, so originally it was?
Yeah.
Yeah, it was brown at first.
It was originally brown. What happened? We stuck it out in the middle of a bay with a whole bunch of oxygen and water around it. What happens to a penny if you let it sit too long? Turns green. Same reason. Because what's happening is the copper is getting oxidized. So it all comes back to who cares, this is chemistry, right? The reason that we care about this in biology is because oxidation reduction reactions can be used to produce energy intermediaries, like ATP. So it's all about moving that energetic electron. A new energy intermediary I want to talk to you about is NADH. So NADH, just like ATP, is produced through exergonic reactions. And it's used to donate electrons, donate energetic electrons to other reactions. Makes them happen. So if you think of ATP and NADH as the energy currency of the cell, NADH is a quarter. ATP is $1. ATP has more energy associated with it and can do more things. But there's some times where you just need a little bit of energy. So you don't want to waste that whole ATP. You don't want to break $1. Nobody wants change. Does that make sense? One of them has higher energy than the other. Both do roughly the same thing. Both power reactions. Both move energy from one point to another. It's just ATP has a higher level of energy than NADH. So that is catabolic reactions, breaking things down. Now we'll talk about anabolic reactions, building things up. So whereas catabolic reactions are going to break down and release energy, anabolic reactions are going to require energy. So where catabolic reactions are exergonic, anabolic reactions tend to be endergonic. Now I'm not going to go into much detail about this because really it's just catabolic reactions backwards. So if you understand how oxidation reduction works, it works the same way with this. That's all I have to say about that. Catabolic and okay, so if you took all of the catabolic reactions and all of the anabolic reactions, you put them together, what do you get? Your metabolism, right. What is an example of an endergonic reaction? Hey, Sarah. A condensation reaction. What's an example of a catabolic reaction then?
Not condensation.
Not condensation. Hydrolysis.